So, hello students, welcome back to CBSE and NCERT Solutions by Vedantu. On this channel, we make solutions and to all the given questions in your NCERT, and at the same time, we also do the NCERT line by line, so that you do not miss out any line of NCERT where you could have a doubt, right? So, this is the second part of the chapter Photosynthesis in higher plants. In the previous class, yes, we finished. In the previous class, we finished the part one. Now in the part 1 we finished starting off with in the entire light reaction. It could be the starting of the photosynthesis, the early experiments and everything. And we also did the entire light reaction which includes cyclic and non-cyclic both. Now if you remember students, this was your non-cyclic photophosphorylation and this right here is your cyclic photophosphorylation. Now if you notice students, a lot of questions will directly come from cyclic and non-cyclic. But students, if you remember, in your non-cyclic and cyclic, did we mention that there is formation of ATP? Yes or no? Quickly tell me in the chat. In your cyclic and non-cyclic both, did we mention anywhere that there is formation of ATP and NADPH? Your answer would be yes, obviously. If you look at your non-cyclic photophosphorylation, and if you look right here, if you look right here, there is a formation of ATP here, and there is a formation of NADPH. So in your non-cyclic, there is a formation of ATP first of all, and at the same time there is a formation of NADPH. This is in the case of your non-cyclic photophosphorylation. Non-cyclic. Now, what about in the case of cyclic? Students, if you remember students, in the case of cyclic photophosphorylation, there was no NADPH. There was no NADPH. But did we see the formation of ATP? Yes, we saw the formation of ATP right here. Can you see? There is a formation of ATP right here in the cyclic. Now, a question might arise. How is the ATP being formed? Explain how is the formation of ATP happening in the chloroplast? Because we know usually ATP is either produced in cytoplasm, that is glycolysis, or it is produced in mitochondria, that is electron transportation and Krebs cycle. But how is it possible to obtain ATP from the chloroplast itself? To explain this phenomena, right, how the ATP is produced, to explain this phenomena, how the ATP is produced, you need to understand something called as chemiosmotic hypothesis. Ready, everyone? Hello, Tanuja. Hello, hello. You need to understand something called as chemiosmotic hypothesis. So, chemiosmotic hypothesis basically explains all it tells you that how ATP is produced. That's all. That's all is the main aim of this hypothesis. It is explaining how the ATP is being formed. For example, let's read the NCERT. You will understand better. Okay, let's read here. Let us now try and understand how actually ATP is synthesized. How actually ATP is made basically in the chloroplast, inside the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism of how the ATP is being produced. Like in respiration, in photosynthesis too, ATP synthesis is linked to the development of proton gradient. For example, students, if when you learn your respiration plants chapter, right, the RIP chapter, when you learn the RIP chapter, you will learn something called as electron transport system or electron transport chain. Now, students, in that chapter, you will understand how ATP is made. For example, you have something called as matrix, we have inner membrane, and we have intermembrane. Intermembrane. You will see H plus coming here and H plus going back again in the presence of something called as F naught F1 complex. Now don't worry students, you will understand all of this in the next chapter, that is respiration plants. But right now the only thing you should know that there is a similarity. There is a similarity between how ATP is made both in respiration and photosynthesis. What is the similarity students? The similarity is going to be proton gradient. In both the cases, the ATP, the main way the ATP, the basic principle behind the ATP formation is proton gradient. I will teach you right now what is the proton gradient, don't worry. Okay, across the membrane. This time, these are membrane of thylakoid. Okay, let's understand electron transport chain with the help of a diagram first of all. With the help of a diagram. See here. Before we start the chemiosmotic hypothesis, before I start explaining, you need to know there are four requirements. There are four basic requirements for chemiosmotic hypothesis to function. There are four main requirements for this to work. What are the four main requirements? The first one is going to be the intact membrane. Can you see? The first one is going to be intact membrane. So we need a intact membrane. Okay, the second one is going to be the proton pump. Proton pump to pump the protons basically. The third main requirement is going to be, can you see this? F naught, F naught, F1 complex. Can you see this? Yes. So we need a F naught, F1 complex, which is also called as what? It's also called as ATP synthase. ATP synthase. ATP synthase. Till here, I believe it's clear. Four main requirements. There are three is mentioned. The last main requirement is going to be the proton gradient. Proton gradient. What is proton gradient now? Understand what is proton gradient now? Proton gradient. Now, all of you know what is proton? H plus. H plus is the proton. Gradient means difference. Proton gradient means difference. Now, what we are trying to do here is we are trying to make a difference in proton level. For example, we are trying to make inside the stroma low H plus concentration, inside the stroma low H plus concentration, and at the same time inside the lumen we need high H plus concentration. That is called as proton gradient. Difference in H plus concentration. In the stroma we need low H plus, in the lumen we need high H plus. But understand what is this lumen, students? Where is the structure actually? What is this structure? All of you remember the structure of chloroplast? In the structure of chloroplast, I told you there are round disc-like structures, if you remember. There are round disc-like structures connected by stromal lamella. Yes. This right here is the magnification of one of the disc. Okay. This right here, this entire disc you see, right? It's a magnification of one of the thylakoid. This right here is a thylakoid membrane. Now students, is this familiar to you? Photosystem 2, photosystem 1, plastoquinone, cytochrome B6F complex, plastocyanin, FNR, FFD, and NAD+. Is this familiar? Have you seen this somewhere, anyone? Have you seen this somewhere? Yes, we have, students. If you go back to your non-cyclic photophosphorylation, which is happening on the thylakoid, see, photosystem 2, plastoquinone, cytochrome B6 complex, plastocyanin, photosystem 1, FES, FD, FNR, NAD+. Same thing is mentioned right here. Do you see? The same thing is mentioned on the thylakoid membrane. Thylakoid membrane. Now, students, now I will teach you one more concept. For this particular chemiosmotic hypothesis to function, I told you there are four main requirements. The first one is intact membrane, it's already there. Proton pump, it is already there. F naught F1 complex, it is already there. The last main requirement, the last puzzle in this piece is nothing but the proton gradient. Now you need to understand how this proton gradient is formed. How this proton gradient is happening. For example, how are we making H plus concentration low on the outside? At the same time, H plus concentration inside the lumen high. How is this proton gradient happening? Ready? Ready, students? To understand how this proton gradient is actually forming. Now, for you to understand the proton gradient difference, how the gradient is forming, there are three main ways. There are three main ways in which you can achieve this, or rather, the cell achieves this. Okay. The first method, students. First method, I'll tell you. If you look at your non-cyclic photophosphorylation, right? If you look at your non-cyclic photophosphorylation, do you notice here there is a continuous supply of H plus? Right, all of you remember in the non-cyclic photophosphorylation, also called as Z scheme, there is complete unlimited supply of electrons here. Electrons are coming. Even though electrons are being used up here, even though the electrons are being used up by your NAD plus, NADP plus, electrons are still coming. From where? What is the magical source here? The magical source of your hello. The magical source of your proton or electron here is the water which we give the plants. The water which you give the plants, the magical source. Right? It is like Doraemon's pocket. How whatever you want, it will keep on producing. Okay, magical pocket this is. Okay. Now what is happening here? We have something called as water which we are adding to the plants, right? Water we are adding to the plants. From this water, the water is splitting here. And the process of splitting the water is called as what? Photolysis of water. All of you know this. All of you know this. Something called as photolysis of water is happening. Now, when the photolysis of watering, did I mention here? This oxygen is released as a byproduct. All of you remember? Yes, all of you remember. These two electrons were taken up by your photosystem 2 for the electron Z scheme. Yes. But what was the fate of this 2H plus students? What was the source of this 2H plus? Hi Sony, I can see the message. Yeah. What was the source of this 2H plus? Where was it going? I told you here it is used for making ATP. Back then, I told you how to make ATP. All of you believed me, yes sir. Now I will show you how it is actually helping in making ATP. Ready? Students, if you notice, we'll come back to this diagram now. I told you there are how many methods to increase the proton gradient? Three main methods to make the proton gradient. Three main methods. The first method out of the three. The first method out of three is, can you see here? Right here, if you see, we have the oxidation of water happening here. Now, in the oxidation of water, also called as photolysis of water, can you see the H plus here? Yes. This was the byproduct. Now, this H plus is stored in the lumen. It is transferred in the lumen. So, can I tell with the help of this, the first way the H plus concentration is increasing inside? Yes or no? I told hi to everyone. Okay, I am very I tell hi to everyone. Tanuja, everyone. I tell hi to everyone. Very generous teacher, you see. Okay. So first was what? First method is what? Increasing the H plus concentration in the lumen. First method is how? By photolysis of water. Wherever there is photolysis of water or oxidation of water, H plus concentration is increasing inside. Done. Done. Understand the second method now. Now if you go back to this diagram right here, see it's very important students. Everything is connected. If you go back to this particular non-cyclic photophosphorylation, in the last step I told you the final electron acceptor is who you say? Who is the final electron acceptor? Tell me. All of you tell me who is the final electron acceptor in your non-cyclic photophosphorylation? It is your NAD plus. NAD plus is the final electron acceptor. And students, I told you here, when NAD plus was accepting the electrons and it was getting reduced to NADPH2, what is reduction? Gaining of electrons or gaining of protons. It is getting reduced here. Did I not mention it is getting 2H plus from the stroma right here? See here. It is getting 2H plus from the stroma right here. 2H plus from the stroma. So can I tell right now? Can I tell because of that condition, inside the lumen, there is no change for this. Rather, there is no change for the second one. But can I tell for the second main option, since proton is being used from the stroma, can I tell in the stroma H plus concentration is reducing? Think again. If H plus is used for reducing NADPE plus to NADPH2, obviously H plus is being consumed. If it is being consumed from somewhere, from where it is consumed? From the stroma. So technically in the stroma there is less H plus now. Yes, less H plus. Yes, yes. H plus is reducing. Now understand. Now understand. That is the second method. Second method is done. H plus is reducing in the stroma. Third and final method of making the proton gradient. Magical word. Okay. The third method is very simple. The third method is, do you see this plastoquinone right here? Now this plastoquinone is not exactly just a electron carrier. It is also a hydrogen carrier. Now what do I mean by that? For example, students, your plastoquinone generally is PQ. It is mentioned as PQ. Right? PQ is plastoquinone. If you see here also, we have PQ as a plastoquinone here. This PQ right here is also a hydrogen donor. What is the meaning of that? Or hydrogen carrier? Meaning of that is this PQ can take a hydrogen from the stroma and become PQH2. That is, it can get reduced. Can you see here in the diagram? PQ to PQH2. When it becomes PQH2, what happens? The excess of proton is now sent to the lumen. Sent to the lumen. Okay, sent to the lumen. Now from this can I tell 1H plus from the stroma, 1H plus from the stroma is being pulled by your plastoquinone and sent inside the lumen. Yes. So with the help of that, H plus concentration is increasing on the inside and outside on the stroma it is decreasing. So with the help of these three different methods, there is a proton gradient is formed. Proton gradient is formed with these three different methods. Okay. Develop. Sad news for you. So three different ways proton gradient is formed and hence the ATP synthesis can happen. Now understand how ATP is formed. See this part here. High electrochemical protection gradient to low. Now what is happening is inside the lumen do I have high H plus concentration? Yes. Inside the lumen now H plus concentration is high. So what is happening is every time 4H plus is sent through this ATP synthase. Understand the lines. Every time 4H plus is sent through your ATP synthase, one ATP is produced. Every single time 4H plus is sent, one ATP is being made in the process. So that is how, friend, that is how in the chemiosmotic hypothesis there is formation of ATP. That is, there is formation of ATP in your light reaction also. Can you see ATP formation in the cyclic, ATP formation in the non-cyclic also. Non-cyclic also. Understood all of you? Understood all of you? This concept is a little difficult for many students to understand because they try to buy hard this. Honestly, if you buy hard this, it will never get to your head. Understand this concept. Okay. Now, there is one difference though. Here, proton accumulation is towards the inside of the membrane, that is in the lumen. Can you see? In the lumen, we had the proton gradient. In respiration, proton accumulation is in the intermembrane space. See, did we write here? Intermembrane space, right? Written right here. In the intermembrane space of mitochondria, when electrons move through the ETC. So we mentioned the difference. In the case of your photosynthesis, electrons are stored in the lumen. In the case of respiration, electrons are stored in the intermembrane space. Intermembrane space. Okay, intermembrane space. Beautiful. Now let us understand what causes the proton gradient across the membrane. We need to consider again the process that takes place during the activation of electrons in the transport to determine the steps. Now how do we get it? We all of you know already, but let's read it. First method is what? Splitting of water. Splitting of water molecule takes place in the inner side of membrane. The proton and the hydrogen that are produced by the splitting of water accumulate within the lumen. I told you, inside the lumen, high H plus concentration is increasing from here. From here, H plus concentration is increasing. Right. The second one you see. As electrons move through the photosystem, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located on the outside of the membrane, transfers the electron not only to the electron carrier, but to a hydrogen carrier. Who is a hydrogen carrier here? That is PQ. PQ is a hydrogen carrier also. Now, this molecule removes a proton from the stroma. It is snatching one from the stroma and sending it inside the lumen. Electrons. When this molecule passes the electron to the electron carrier on the inner side of the membrane, the proton is released from the inner side or to the lumen side of the membrane. So can you see here? From this plastic muon is sending a proton towards the lumen, increasing the concentration inside. The third one, the NAD plus reductase enzyme is located on the stroma side of the membrane. All of you know that. NAD plus reductase enzyme, FNR enzyme. You're right here. Can you see? The FNR enzyme. FNR enzyme. Okay. The FNR enzyme, now along the electrons that come from the acceptor of the PS1, protons are necessary for the reduction of NAD plus to NADPH2. So in this conversion, what is happening? In this conversion or reduction of NAD plus, one H plus is being used up from the stroma. From the stroma. Okay. Now hence within the chloroplast, protons in the stroma decrease. Very important line. Stroma it is decreasing, while lumen there is accumulation protons. So inside the lumen, we have high H plus is high, And inside the stroma we have H plus is what? Very low. H plus is very low. Hmm, exactly. Simple. Now, why are we student product gradient? This gradient is important because it is breakdown of this gradient that leads to the synthesis of ATP. So, for ATP to be formed, this gradient has to break down. So, when you break things, you can make new things, right? Unless you break something, you can't make new. Very rarely it will happen. When you want to break a bad habit, you will obtain a new good habit. Understanding? If you want to break a bad habit from your life, you need to replace it with a good habit. Unless you replace your bad habit, right, with something new, the bad habit will keep on coming back. For example, if you have a bad habit of not reading NCERT, or you have a bad habit of not solving PYQs, replace with a good hobby. For example, you have a bad habit of procrastination, replace procrastination with a good hobby, and you are good to go. So here is what is happening. For the ATP synthesis to happen, the proton gradient has to break down completely. It has to completely break down. Why is my hair looking so weird? Okay. Now, the gradient is broken down due to the movement of the proton across the membrane to the stroma through the transmembrane channel of F naught of synthesis. So F naught protons are coming out and ATP is made. ATP is made. Okay. The ATP synthase enzyme consisting of the two parts, one called as F naught, other one is called as F1. F1. Okay. Breakdown of gradient provides enough energy for the conformal change of the CF1 particle of the ATP synthase, which makes the enzyme synthesis overall molecular energy packed with ATP. That's all. By mistake, I became a botany lecturer, motivation speaker someday. Let's see. But botany is fun. Botany is really fun. I don't know what else is fun, but botany is actually easy and fun. See, every single line of NCRT is so easy to understand when it is truly easy and very amazing. So I don't think so. I would give a botany to anything else to do anything else. So yeah. Now, chemiosmotic uh, chemiosmosis requires a membrane. I told you all this requirements. It needs a membrane, a proton pump, a proton gradient, and ATP synthase. And ATP synthase. Four main requirements. Okay. Energy is used to pump the electrons across the membrane to create a gradient or the high concentration protons within the allocat membrane. All of you know this already. All of you know all of you know this already. Yes, botany is really fun. Trust me, students. All this, right? Trust me. See, I told you right now we're learning. I'll give you a image of what we're learning right now. Okay. All of you close your eyes and imagine where this is actually happening. All of you. See, all of you close your eyes. Imagine now. Imagine there is a wide forest. In this amazing forest, you have one tree. In that one tree, there are hundreds and thousands of leaves. Hundreds and thousands of leaves. And you take that one leaf, and you inside that one leaf, it is made up of so many different type of tissues. Inside that tissue, there is a cell. Inside that cell, there is a chloroplast. Inside the chloroplast, there is a thylakoid. In on the thylakoid, ATP synthesis is present. In that ATP synthase, ATP is being made. Do you realize what you're trying to learn right now? You're learning this information which you're learning right now is very difficult to access back in the day. But you're fortunate enough to understand all this because it has already been discovered. Okay, so learn the importance of this. Learn the importance of what you're learning right now. It may be just written in your NCERT lines. It may be just written as in your NCERT lines, like randomly to have written. But understand the gravity or the importance of lines of NCERT when I talk about thylakoid membrane. Imagine from a large forest, we are coming to a cell chloroplast on a thylakoid membrane of the thylakoid. We are learning the information on that level. Isn't it truly amazing from what humans have discovered for you to learn such an intricate knowledge? Tell me. Unfortunately, Sony, in the roots, this will not happen. Because roots do not have chloroplast, right? Roots do not have chloroplast. Roots do not have chloroplast, so it will not be happening in roots. But understand what you just are learning right now. It is so important. For example, students, for example, I have this particular plant right here. I have this particular plant right here. In this plant, this dark green region, I have chloroplast there. Inside the chloroplast, I have the thylakoid. On that thylakoid membrane, not even inside, on the membrane, I have non-cyclic photophosphorylation happening right now. Do you believe that? It is happening right now because I kept it in sunlight for this all this while. It is happening, all this is happening right now in a plant. So appreciate what is happening, right? So do you realize what is the importance of such a thing which you are trying to learn? You might be thinking, sir, I'm doing all of this for marks' sake. If I get good marks, I will get into a good college. But it is that is good. That is good. But let me tell you, students, it's always more than that when you're trying to learn botany. Appreciate what you have around you, and you will see the magic behind it. Unless you truly appreciate what you are learning, right? You will simply read for the marks' sake. Don't be that person. Okay? Don't be that person. Now, with that being said, let's continue forward. Let's continue forward. Okay. ATP synthesis has a channel that allows diffusion of proton back into the membrane. This releases enough energy to activate the ATP synthase enzyme and catalyze the ATP formation of ATPs here. This is what they're talking about. Here, students, as a proton, imagine this is actually happening in a cell, in a plant right now. Okay, this H plus right here is passing through the membrane and passing through the ATP synthase. So we have H plus coming here. So when the H plus is traveling here, it makes ATP. It makes ATP. Okay. Exactly. Botany is nature. Appreciate the nature. You will love it. Understanding all of you? No. Along with NADPH produced by the movement of electrons. See, NADPH in the non-cyclic. Remember, NADPH is produced only in non-cyclic, not in cyclic also. All those small details, right, will be asked as question, and it has been asked as question. It has been asked as a question. So remember, NADPH is only produced where? In the non-cyclic photophosphorylation, not in the cyclic, not in the cyclic. Okay. Now, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma, possibly for fixing CO2 and synthesis of sugar. This is basically the introduction of dark reaction. Dark reaction. Yes, proud to be plant. Exactly. Be proud to be plant. All of you are plants. Now, now students, we'll go to the next step now. We'll go to the next step. Now we'll go to the next step. Okay. Next step is what are the ATP and NADP used for? See, ATP and NADPH both are produced in the light reaction. In the light reaction, both are made in the light reaction. Now this ATP and NADPH, now they'll be used in a separate reaction called as biosynthetic phase. Biosynthetic phase. This biosynthetic phase is also called as your dark reaction. Ready? Using dark reaction. On to the next step now. Next step is dark reaction. Okay. Students, should I draw it for you or should we read first? Tell me. Should we draw first or should we read first? I think we will draw the cycle and then understand it better way and then go ahead. No? Let's draw the cycle first. All of you should know this by heart. All of you should know this what is happening every step. Okay. Let's understand dark reaction. Dark reaction. Okay. Dark reaction is also called as your C3 cycle. It is also called as your Kelvin cycle. Important. Three things. It is called as dark reaction, C3 cycle or Kelvin cycle. I'll tell you why in all the things. It's called as a dark reaction because it does not require light. It does not require light. It is called as C3 cycle because the first stable product in your dark reaction is a 3 carbon compound. That is why it's called as dark reaction. It's called as C3 cycle. Why is it called as Kelvin cycle? Because the person who discovered the cycle, his name was Melvin Kelvin. Right? So his name was Kelvin. So we kept it as Kelvin cycle. Okay. Kelvin cycle. Now understand all of you. Understand. I want everyone to 110% attention now. Okay. First of all, let's take one simple this thing, right? First of all, let's take a simple CO2 entering the cell. Now we have a CO2 entering. Let's show CO2. CO2 is entering. Okay. Now this CO2 will be taken up by a will be taken up a 5 carbon molecule called as RUPP. That is nothing but rubulose 1, comma 5. One second, students. Rubulose 1, comma 5 bisphosphate. 1, comma 5 bisphosphate. This RUBP is 5 carbon. This CO2 is 1 carbon. Now you tell me, what is 5 plus 1? 6. So all the scientists in the world, students, all the scientists in the world were also looking for a 6 carbon, like all of you. Okay, mental only at one point. Okay, they became mental looking for it. Okay, so this RUBP right here was technically supposed to give a 6 carbon molecule. But did they get it? No. They got a molecule called as PGA. PGA. That is phosphoglycerate. 3 phosphoglycerate. 3 phosphoglycerate. Okay. 3 phosphoglycerate. Okay. 3 phosphoglycerate. PGA. Now this is how many carbon? 3 carbon. Now what if I make into 2 of it? Tell me now. 3 into 2 is how much? 3 2s are? 3 into 2 is 6 carbon. So we get 6 carbon here and we get 6 carbon here. So 6, 6, 6, 6 matching. So this step, in the first step, is actually called as carboxylation. This is called as carboxylation. The first step is called as carboxylation. Okay, in which carbon is being fixed. Carbon is being fixed. In this first step, there is also an enzyme, friends. Enzyme called as Rubisco enzyme. Bacillin Rubisco. Rubisco enzyme. Rubulose 1,5, carboxylase and oxygenase. C is carboxylase, O is oxygenase property of Rubisco. Okay, carboxylation done. Now students, in the second step what happens is, now this PGA, right, this PGA will now be converted to one more structure called as triose phosphate. Triose phosphate. Now this triose phosphate is 3 carbon molecule again. This triose phosphate will eventually get converted to glucose. Now second step is called as your reduction. Called as reduction. Now the second step reduction is not free of cost, because the second step there is some things are happening. That is, for example, 2 ADP, 2 ATP is used up here. 2 ATP is being converted to 2 ADP. So can I tell 2 ATP is used here? Yes. At the same time, the second step only, we have NADPH2 is getting converted to NADP plus. So can I tell 2 of each also can consume? So in the reduction step, the second step, 2 ATP and 2 NADPH is being used. 2 ATP and 2 NADPH is being used in the reduction stage. The last stage is called as regeneration. From triose phosphate, again we are converting back to, not we, plants do it actually, very smart plants. Okay, why is this color? India flag it became. Now what is the third step? In the third step, called as regeneration. Third step is called as regeneration. Your triose phosphate is again getting converted back to, triose phosphate is again converted back to RUBP. Again in this step also, 1 ATP is being consumed. 1 ATP is converted to 1 ADP. So this is the 3 steps of your dark reaction. That's all. Nothing more than this. Dark reaction is over. Moto ads are back. I can't control the moto ads. I'm sorry. Understanding all of you? So what we learn right now? C3 cycle is over now. Okay. First step is happening. What is happening? Rubisco RUBP plus CO2 will give you PGA. PGA will go convert to triose phosphate. In the process, there are 2 ATP and 2 NADPs are used. The triose phosphate is being converted back to your RUBP. In that step, again 1 ATP is being consumed. ATP consumed. First step is called as Rubisco. Enzyme is first step is called as carboxylation. Enzyme is called as Rubisco. Second step is called as reduction. Third step is called as regeneration. That's all. C3 cycle is over. C3 cycle is over. Sir, how to avoid ads without taking premium? Use Brave. Brave. It's a Brave is a this thing like Google. What do we call that? I forgot what we call that. It is a search engine. Brave is a search engine. Try downloading Brave. If you download Brave, there will be no ads on YouTube. If I remember correctly last time. Or you can also buy YouTube Premium, which is around 50 rupees per month or something. 50 or 70 rupees per month. YouTube Premium. Right. Those are the two only two. Or you can. That's all I think for avoiding ads. Otherwise, there is no other go. See, students, you need to understand even if our Vedantu channel doesn't put ads on the video, YouTube will put the ads on the video later on. Yeah. So what do we learn right now? We learned right now here is for one CO2, how many ATP and how many NADH is used now? Tell me. For one CO2. To fix one CO2, we need totally three ATPs, right? Two here and one here. So three ATP. At the same time, there is also requirement of two NADPH. Two NADPH for one CO2. In examination, they can ask you, what about for glucose then? What about for glucose? Tell me. If for one CO2, this much you need, how much will be required for 6 ATP, 6 carbon? Tell me. All you need to do is 3 ATP into 6 and 2 NADH, NADPH into 6. 18, 18 and 12. Yes. 18 and 12. Tell me. Tell me, is it right? Tell me, is it right? 18 and 12. That's all. That's all is the thing. 18 ATP and 12 NADPH. This is a famous question. Last year NEET exam which they asked you. 2015 they have asked. 2022 they have asked. So this is a famous question. How many ATP and NADPH is for one glucose? That's all. Done. C3 cycle is done. Now let's read what is given in NCERT now one more time to confirm whatever we have read is making sense or is it true? Okay. Now let us now see how ATP and NADP is used in biosynthetic phase. ATP is used and NADP is used in biosynthetic phase. Earlier we saw that CO2 is combined with H2O to produce the glucose. This first reaction. Okay. Now, or sugar. It was interest to scientists to find out how this reaction proceeded, or rather, what was the first product of your CO2 is taken into reaction or fixed. The first product was what? The first product was your PGA as the first product. Since it is three carbon, that's why it's called a C3 cycle also. Remember that, okay? Remember that. Now, just after World War II, among the several efforts to put radioisotopes to beneficial use, the word of Melvin Kelvin, which is nice name, no? Melvin Kelvin. Hmm. Okay, Melvin Kelvin. Okay, Melvin Kelvin is exemplary. The use of radioactive 14 carbon by him in algae photosynthesis studied that discovery led to the CO2 fixation product was a 3 carbon organic acid. See, 3 carbon organic acid, that is PGA. He also contributed to working out the complete biosynthetic phase, it's called as Kelvin cycle. After him, the first product to identify was PGA, okay, also called as PGA, 3 phosphoglyceric acid. Now, scientists also try to know whether all plants have PGA as the first product of CO fixation. So they understood some plant is making 3 carbon compound as the first product, but some plants are making 4 carbon compound. So whatever plants are making a 3 carbon first product, they call them C3 cycle, C3 plants. Or some plants which were making a 4 carbon product, that we call them C4 plants. And the plants that are making 4 carbon were called as oxaloacetic acid. This was the 4 carbon C4 carbon product. Okay. Yes, Kelvin Melvin is like gingerly oil. Gingerly oil, trust me, students, I can never keep a straight face when I'm teaching biomolecules, and especially when I reach the fat lipids. In lipids, there's something called gingerly oil. Okay, gingerly oil. Gingerly oil. Right. So, yeah. So, understand gingerly. And tell me, students, let me know in the comment section today. Let me know in the comment section today. Where do we understand the example of gingerly oil? Where do we study the example of gingerly oil? Is it in saturated or unsaturated? Not here. Not here. In the comments later on after the video, tell me where do you study gingerly oil? Do we study that under saturated or unsaturated? Okay. Time there later on. This is the homework for all of you. Small homework from biomolecules chapter. Now, those of you wondering, sir, where do I study biomolecules? Those of you know, know it. Okay, from where you study biomolecules. Okay. Now, since CO2 assimilation during photosynthesis was said to be two main types. One is called a C3 and one is called a C4. Very simple. What is gangrene oil? No, that is not gangrene, it's gingerly oil. Gingerly oil. You will study that in biomolecules, class 11th chapter, not class 12, that is chemistry. Okay, clear? Gingerly oil. Who keeps the name gingerly oil? It is so funny. Okay. The primary CO, acceptor of CO2, all of you know. Tell me, students, who is the primary acceptor of CO2? When the CO2 was lonely and crying in a corner, who came to help our CO2? Tell me. Saturated haloalkanes. No, you don't tell me. Tell me. It is haloalkane. Yeah, makes sense. Okay. Tell me. When CO2 was lonely, who accepted CO2 in its house? I mean, who is that 5 carbon compound is like, please come inside my house and take rest here. That was your, tell me, it is your RUBP. RUBP. Okay, RUBP. Now they also believed that since the first product was a 3 carbon, the primary acceptor would be a 2 carbon. It makes sense. It makes sense. Right? If product is only 3 carbon, CO2 is 1 carbon. See, if product is going to be 3 carbon, CO2 is 1 carbon. Obviously, the first acceptor is supposed to be 1 carbon, right? 1 carbon. So it's 2 carbon. Why? Because 2 plus 1 is going to be 3 here. That's what they thought, students. Scientists also very simple thinking they did. Scientists also did very simple thinking. Right? 2 plus 1 is 3, and the first product is going to be 3 carbon. So this reactant is supposed to be 2 carbon. But surprise, surprise. What a surprise. The first acceptor was 5 carbon. CO2 is 1 carbon. So we ended up getting 6 carbon as a 6 carbon molecule. That 6 carbon is nothing but 2 times PGA is the 6 carbon. Understood all of you? Okay. No. The primary acceptor is 2 carbon. They spent many years trying to discover the 2 carbon compound before they discovered the 5 carbon RUBP. RUBP is finally found. Kelvin cycle. Okay. Very important line here. Let us now outside the understanding very clear that Kelvin cycle pathway occurs in all photosynthetic plants. What is the meaning here? Your C3 cycle will happen in your C3 plants. C3 cycle will also happen in C4 plants. Both the plants C3 cycle will take place. Now how come, sir? C4 cycle is called C4 cycle because it has C4 cycle, right? But let me tell you, students, when I teach you C4 cycle today, when I teach you C4 cycle today, you will understand. Because in C4 cycle, C3 is also happening. Okay, so we'll come there. We will come there, don't worry. Okay. Now let's read what is given. It does not matter whether they have C3 or C4. Okay. For ease of understanding, let's Calvin cycle can be described as the following. First one is called as carboxylation, all of you know, where CO2, right? Where CO2 is stable organic intermediate. Carboxylation is the most crucial step of Kelvin cycle where CO2 is utilized for carboxylation of RUBP. So RUBP plus CO2, right? This reaction is catalyzed by the enzyme RUBP, carboxylase and oxygenase. What is the meaning of that? Rubisco. Rubisco. Okay, and we get two molecules of PGA, two molecules of 3 PGA. Since the enzyme is also called as oxygenase, we can call it Rubisco enzyme. Okay, Rubisco enzyme. Now, second step. These are a series of reactions led to the formation of step of glucose. So, in reduction, glucose formation is happening. This step involves two molecules of ATP used up. Two molecules of ATP gone. Two molecules of NADPH is also gone, used up in the reduction step. Okay. Now, the fixation of 6 carbon, that is CO2, 6 turns of the cycle are required for formation of 1 mole of glucose. That is, for one time the cycle is happening, right? That is happening for one CO2. For if you want to make it for glucose, 6 times the cycle has to go. The cycle has to go 6 times to make one glucose. Okay, to make one glucose. Last step is regeneration. Very simple again. What do we learn in regeneration, students? In regeneration is what is happening. Your triose phosphate is getting converted back to RUBP with the use of 1 ATP. That's all. Now, regeneration of CO2 acceptor molecule RUBP, crucial if the cycle has to continue. The regeneration of step requires 1 ATP phosphorylated to RUBP. That's all. Last step. RUBP formation. So with that, your C3 cycle is over actually. See here, we learned the entire thing. RUBP, CO2 is entering, carboxylation, we get two molecules of PGA, 2 ATP and 2 NADP is used in reduction. Triose phosphate gets converted to glucose later on. In the next step, regeneration, 1 ATP is used to make 1 ADP, and finally we get RUBP back. Easy, right? Tell me, easy or not? With that, C3 cycle is over. That's all is the C3 cycle. Very, very simple cycle. C3 cycle is very simple to understand. See here, this is what given for glucose. For glucose, 18 and 12. Did we learn this? 18 and 12. Did we learn this? 18 and 12. I told you no. 18 and 12. Yes. 18 and 12. 18 and 12. Now, with that, we come to the next part of the chapter that is called as photorespiration, also called as C2 cycle. Also called as C2 cycle. Ready? Tell me, ready, all of you? Ready for C2 cycle? Ready for C2 cycle? And all of you who are feeling sleepy, go drink some water. All of you who are feeling sleepy, go drink some water. Quickly go drink some water. Tell me if you're ready for C2 cycle now. All of you. Ready? Just my chair. Okay. You said some of you are ready. So let's start C2 cycle. Now students, first I'll teach you C2 cycle my way. And then we'll read NCERT. Ready? Ready all of you? Okay. Understand students. Understand a simple thing you first understand. All of you remember Rubisco? All of you remember Rubisco from previous experiment or previous C3 cycle. This C is called as carboxylase, O is called as oxygenase. C is called as carboxylase, O is called as oxygenase. Now this Rubisco can form a bond with CO2 or it can form a bond with oxygen. It has a choice. This particular Rubisco has to make a choice if it wants CO2 or if it wants oxygen. Okay, what wants? Exactly. Water is very important. It needs oxygen. Now, this Rubisco right here will always choose CO2 when you say when CO2 and oxygen levels are same in the atmosphere. In the atmosphere. So when CO2 and Rubisco, when both are same level in the atmosphere, Rubisco will always go for whom? CO2. Always go for CO2. But as soon as as soon as oxygen levels increase in the atmosphere, if oxygen level is increasing in the atmosphere, Rubisco will leave CO2 happily. Full betrayal, Katappa it became. It betrays CO2 and forms a bond with oxygen. Forms a bond with oxygen. Similarly, if what will happen if CO2 will decrease again? Tell me. If CO2 is also decreasing, again Rubisco is forming a bond with oxygen. With bond with oxygen. So Rubisco is ready to leave. Rubisco is ready to leave CO2 at any point in its life. Every single time CO2 goes down, it'll be like, bye bye, tata, bye bye, CO2. I'm going with oxygen. If oxygen increases, again Rubisco will say tata, bye bye, CO2. I'm going with oxygen. Rubisco will only be present with CO2 when they are equal or CO2 is very more compared to oxygen. Only in those conditions, Rubisco will be with CO2. Full opportunistic now CO2. Very cunning it is. Wherever it sees benefit, it will run behind that. Rubisco will run behind the benefit always. Ready? All of you understood? Now understand the process of fluid respiration C2 cycle. I will draw for you, then understand C2 cycle. C2 cycle. What is happening in C2 cycle first of all? First of all, we have something called as RUBP again. Now, in this case, RUBP is forming a bond with oxygen, not CO2. So, RUBP is 5 carbon. Oxygen is how many carbon? 0 carbon. So, technically, we are supposed to get 5 carbon here. So, we don't get an entire 5 carbon product. We actually get two products. One is called as PGA, which already, if you know already, is 3 carbon plus PG. PG is going to be 2 carbon. This PG is called as phosphoglycolate. Phosphoglycolate. Phosphoglycolate is produced. This is called as your C2 cycle. This is called as C2 cycle. But the problem here is very big problem for the plants. What are the main problem here? The main problem here is that this reaction, this entire cycle, no sugar formation, no glucose formation. At least in C3 cycle we had glucose formation, right? At least in C3 we had glucose formation, but in C2 no glucose. No ATP is being synthesized or no ATP is being produced here. Not being produced. Nothing is happening. ATP is not being produced here. Okay. And ATP is being consumed. ATP is being used in this process. Okay. Fourth one. CO2 is not used. Can you see? There is nowhere CO2 is used. So if CO2 is not used by the plant, can I tell the CO2 is escaping through stomata? Yes. CO2 is escaping. CO2 is escaping. So if there is no sugar, ATP is being consumed, CO2 is being released. Can I call this a waste body? Waste process? Can I call this a waste process? Completely waste of time for plants? Yes. So now you might ask me, sir, so if this is completely useless to the plants, why are they even doing it? What is the main reason they're doing it, students? The main reason they're doing it is because of the atmospheric changes. If there's a change in atmosphere, they have to do it. Papa, the plants do not have an option. That is, C3 plants do not have an option. C3 plants do not have an option. It has to undergo C2 cycle. It has to undergo C2 cycle. There is no option at all. Mhm. So the sad fate is always with the C2 cycle. Okay. Now, let's see what is given in NCERT though. See, let us try to understand one more process that creates a difference between C3 and C4. That is photorespiration. Again, photorespiration difference between C3 and C4. Why is the difference? Because C4 plants, this particular reaction, that is fifth point here, right? C2 cycle will never happen in plants. It will never happen in plants. Never happen in plants. In where? In your C4 plants. C4 plants are very smart plants. C4 plants are very smart plants. So C2 cycle will never happen in your C4 plants. But Papa C3 plants has to go through this. They don't have an option. C3 plants don't have an option. C4 plants very smart plants. They are like, whatever you do, nothing will affect us. That is C4 plants. Okay. Let's read what is given in NCERT. I hope all this storytelling is helping all of you, right? I hope it is helping all of you. Okay. I hope this is helping all of you to understand the wastefulness of your C2 cycle. Okay. To understand photo respiration, we need a little bit of first step of carbon. See, all of you know, this is the first step, right? This is the first step of your carboxylation cycle. Now, Rubisco, that is almost most abundant enzyme in the world, obviously. PYQ again. Rubisco has a much greater affinity for CO2 when they are equal. When they are equal, same. They'll treat very equally. Example, I'll tell you classic example here, okay? Here we have one friend. Let's call him bot. Okay. Let's not call him bot. I am bot. This is not bot. Okay. Let's call this person as Dino. Okay, his name is Dino. This specific person Dino, whenever this A friend has more money, he'll go with him. The same Dino, when B friend has more money, he'll go with them. This A friend is like CO2. B friend is your oxygen. And O, this Dino, other name of Dino is nothing but Rubisco. Rubisco will go behind CO2 when CO2 is more. But when oxygen is more, Rubisco will go behind oxygen. Like how Dino will go behind who has more money, no. Rubisco will also go behind wherever is necessary. Wherever is necessary. Sad story, this is one sad story, understand. Sad story is happening. Okay. Let's erase this story before it gets viral. Before it gets viral. Erase, erase, erase. Okay. Now, where were we now? See here. Imagine what would happen if there was not so. This binding is competitive. It is relative concentration of O2 and CO2 that determine. In CT plants, some O2 bind with Rubisco, hence its CO2 fixation is decreased. Okay, the same thing they're telling. What have we learned right here? Okay, food respiration. That is, here RUBP, instead of converting in two molecules of PGA, binds with oxygen. See, RUBP binds with oxygen to form one molecule of phosphoglycerate and phosphoglycolate. Two carbon. Phosphoglycolate. In this pathway called as photorespiration. Okay. So it is a wasteful process. It is a wasteful process. Now, students, do you want to know? Do you want to know how C4 is overcoming this? Story time again. I'll tell you one more story now. Tell me, do you want to know how C4 plants were very smart plants? Your C4 plants were very smart plants. How are they overcoming this barrier? Like, how will they make sure? How will they make sure that Rubisco is never finding oxygen? Want to know? That is the story for C4 cycle. Want to know how your C4 cycle are making sure that your Rubisco is never forming a bond with oxygen? Let me tell you. Let me tell you. Very simple, students. Very simple. See, for plants are basically making a jail. They're making a small room here, like a jail room. In this small room, CO plants are sending the CO2. But who is waiting here? Guess what? Who is waiting in this room? In this room, the only enzyme present here is Rubisco. And they have made it in such a manner that oxygen cannot enter here. So you tell me, students, you tell me, understand here, if your oxygen is not able to enter inside, and in this room right here, you only have CO2 and only have Rubisco. Tell me now, this Rubisco does not have any option. It has to only bind with CO2. So tell me, is there any way for photorespiration now? Is there any way that this Rubisco will form a bond with oxygen and make a PG, right? Phosphoglycolate. So can I tell in your C4 plants were so special, they're making sure in that small room, right, your oxygen is not entering inside. Make sure that oxygen is not entering inside and CO2 and Rubisco are present all alone and sugar is being produced. Clear all of you? Clear all of you? Clear? This is what is happening inside the C4 plants. C4 plants. Now, let me teach you C4 cycle, then we'll read what is given in NCERT. Let me teach you C4 cycle. To understand the C4 cycle, you need to know something called as Kranz anatomy. Because if you take a C4 plant leaf and cut the leaf, you will get such a structure. If you look at the Kranz anatomy, obviously there is something called as upper epidermis, lower epidermis, which is also present in a normal leaf. Upper and lower epidermis is normally present in all the leaves. Okay. Now here what is happening here is this inside part. You see this inside part is the vascular bundles. Vascular bundles. Can you see around the vascular bundles there is a wheel-like structure. This wheel-like structure is called as wreath, and this wreath is made up of bundle sheath cells. So if you imagine this one round thing here, that round bundle sheet is completely covered from all the sides by the bundle sheets. The bundle sheet is the small room which I was telling you, in which oxygen cannot enter. Around this bundle sheet, on the outside also there is one more round like structure. It is called as mesophyll. So on the outside we have what? On the outside we have the mesophyll, and on the inside we have the bundle sheet. Inside we have the bundle sheet here, bundle sheet. And bundle sheet is very tightly packed. Why? To make sure oxygen is not entering anywhere. It is tightly packed and it has a high amount of chlorophyll or high chloroplast, large chloroplast in the bundle sheet. So we have bundle sheet here. This is bundle sheet, and outside part is called as what? Outside part is called as mesophyll. It is called as mesophyll. Okay, mesophyll on the outside. This is called as Kranz anatomy. This is called as a wreath. See, similar structure, no here. This part, this red part. That is called as wreath-like structure or bundle sheet. Okay. Now understand C4 cycle, then we will read everything what is given in NCERT. Understand it. It's very beautiful, very simple to understand. Okay. For example, we have two cells I told you here. We have two different cells here because the reaction is happening in two separate rooms. Now understand what is happening. Now as usual, CO2 is entering. Now question will be asked, CO2 is entering which cell? For that you need to know which cell is this. This cell is called as mesophyll cell. Mesophyll cell. And this cell right here is called as bundle sheet cell. Bundle sheet. The same bundle sheet I told you, wreath-like arrangement, tightly bound. CO2 is entering. This CO2 is picked up by. The CO2 is picked up by. See, in the case of your C4 cycle, in the case of C3 cycle was RUBP. RUBP was taking it, but here you have something called as PEP. What is this PEP, students? PEP is nothing but phosphoenol pyruvate. Phosphoenol pyruvate. Phosphoenol pyruvate. Now this is one carbon. This phosphoenol pyruvate is 3 carbon. Tell me what is 3 plus 1? 3 plus 1 is a 4 carbon compound called as OAA. OA is nothing but oxaloacetic acid. OA is what? Oxaloacetic acid. Oxaloacetic acid. This is 4 carbon. That is why this is called as C4 cycle. Also called as Hatch and Slack pathway because discovery was done by Hatch and Slack. That will be tell later on. Now this OA is converted to malic acid. Malic acid. Now this malic acid now will be sent into the bundle sheet. So what we have here? We have the malic acid. Malic acid is converted to OA. OA is converted to pyruvate. Pyruvate is 3 carbon. OA is 4 carbon. 4 to 3. Can I tell one carbon is escaping somewhere? That is right here. One carbon is escaping. Now this carbon is now surprisingly picked up by none other than your RUBP. Again RUBP is coming. Now this RUBP is taking the CO2 and in the presence of enzyme called as Robisco enzyme, we are getting the 3-PGA. 3-PGA is again converted to triose phosphate to RUBP. So can I tell in the bundle sheet, technically in the bundle sheet we have the C3 cycle going on? Yes or no? All of you in the chat, tell me. In the C4 cycle, we have the C3 cycle also going on right here in the bundle sheet. So that's why they tell C3 cycle is present in all the plants, be it C4, be it C3. Okay. This pyruvate is again sent up. Pyruvate. Now this pyruvate is now converted back to PEP. It is again converted back to PEP. Okay. Pyruvate is again converted back to phosphoenol pyruvate with the using of 2-ATP. 2-ATP is used in this process to make 2-ATP. Pyruvate is converted back to PEP. Now here you might ask me, sir, here you had Rubisco enzyme. What about any enzyme in the mesophyll cell? Yes, there is an enzyme here. In the mesophyll cell, the enzyme is called as pep carboxylate right here. PEP CASE, pepcase or pep carboxylase is the enzyme. Pep carboxylase is the enzyme. Understood all of you? I will repeat again, all of you just listen to me one more time. First of all, we have the mesophyll cell, bundle sheet cell. CO2 is entering. CO2 is taken up by PEP. 3 carbon, 1 carbon, 4 carbon compound, also called as C4 cycle, oxaloacetic acid. Oxaloacetic acid is converted to pepcase. And this process of PEP to OA, the enzyme is called as pepcase. And pepcase will be only present in the mesophyll, not in the bundle sheet. Not in the bundle sheet. Pepcase will be found only in the case of mesophyll cell. Only in the case of mesophyll cell. Okay. Now, malic acid. Malic acid is sent into bundle sheet. Malic acid converted to oxalic acid. Oxalic acid converted to pyruvate. 3, 4 carbon, 3 carbon. 1 carbon is missing. That has been removed here. Now, this enters the C3 cycle in the bundle sheet. CO2 will enter the C3 cycle. Now, this pyruvate is sent up to mesophyll cell again, where the pyruvate gets converted to PEP again. 5 for the reaction to keep on happening to PEP. And this step requires 2-ATP. This step requires 2-ATP. That is the story of C4 cycle. Done. 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 That is the cycle of your C4 cycle of story of your C4 cycle. Okay. And what we learn here, students? What we learn? What did we learn? What did we learn? Let's read the NCERT now. In C4 plants, in your C4 plants, in C4 plants, food respiration does not occur. All of you know this already. All of you know this already. Does not happen. This is because they have mechanism that increases the concentration of CO2 at the enzyme site. This is called as your bundle sheet cell. Bundle sheet cell. In this bundle sheet, we have high CO2 and enzyme is present here. That's what they're telling you here. This takes place in the C4 acid. When the C4 acid from the mesophyll cell is broken down in the bundle sheet to release the CO2, that is oxaloacetic acid is broken down to give pyruvate. This resulting increase the intercellular concentration of CO2. In turn, this ensures that Rubisco functions as carboxylase, minimizing the oxygenase activity. Now that you have, now you know that the C4 plants lack food respiration, you can probably understand why the production yield are much better in these plants. Obviously, there is no food respiration. If there is no wasteful process C2 cycle happening in your C4 plants, productivity of your C4 cycle is very high. Productivity is very high here. In addition, these plants show high tolerance to high temperature and high light. The plants that adapted to dry tropical regions have C4 pathway. C4 oxaloacetic acid as the first product. As CO2 fixation products are used for C3 pathway of or Kelvin cycle. Okay. They are very special. Why? Special type of leaf anatomy. They are tolerate high temperatures. They show response to high light intensities. Lack process called as photorespiration. More productivity in C4 plants. C4 plants are special. They have special type of leaf anatomy. Okay, that is called as Kranz anatomy, where vascular bundles of CO plants are called as plants. The vascular bundles of CO plants are called as bundle sheet cells, and leaves which have such anatomy is called as Kranz anatomy. Kranz means breadth. The reflection of arranged leaves, the bundle sheets are may form several layers around the vascular bundle. They form many layers. Bundle sheet forms many layers. They are characterized by having a large number of chloroplasts, thick wall, impervious to gas exchange. That's why they say oxygen cannot enter inside the bundle sheet. Cannot enter. Okay. Example is given here. Maize and sorghum are the examples of C4 plant. Example of C4 plant. Maize and sorghum. Okay. Now it would be interesting to for you to collect the leaves of diverse species around you. Basically, they're trying to find C4 cycle. Now, what is happening in C4 cycle? CO2 is acceptor is a 3 carbon. I told you. Phosphoenol pyruvate. When PP reacts with with CO2, we end up getting OA. We end up getting oxaloacetic acid. Is it enzyme here? Yes, it has enzymes. The enzyme here is what? Pep carboxylase is the enzyme. Now tell me, do you find this pep carboxylase in the mesophyll or in the bundle sheet? Tell me. Obviously, students, where do you find pep carboxylase? Pep carboxylase is only and only found in the above cell. That is mesophyll cell. It will be found only in the mesophyll cell. Only in the mesophyll cell. Pep case. Pep case can never be found in bundle sheet. Okay, bundle sheet. No. OA is found. The four carbon compound like malic acid or aspartic acid in the mesophyll cells itself. See, all of you know malic acid we have taught you. These C4 acids are broken down to release CO2, a three carbon molecule. This is pyruvate. So malic acid or oxaloacetic acid is broken down to pyruvate to release one CO2. The CO2 released in the bundle sheet are entering the C3 cycle, where C3 cycle is happening in the bundle sheet cell. The bundle sheet cells are rich in Rubisco enzyme but lacks pepcase. Okay, that is your C4 cycle. That is your C4 cycle, students. Thumbs up, taste of thunder. Some jokes you're telling. Okay, understood all of you? C4 cycle is done. Any doubt in C4 cycle? See here, one more time I'll tell you. Here we have phosphoenol pyruvate, PEP. CO2 is coming. CO2 fixation will happen. We get a C4 acid. That is nothing but your malic acid. That is nothing but your oxaloacetic acid. Transported here. Malic acid. Malic acid undergoes decarboxylation. So one CO2 is removed and we get a 3 carbon. That is pyruvic acid. Pyruvic acid again transported up on top. But this CO2 enters the C3 cycle here. C3 cycle here. This undergoes regeneration to make again phosphoenol pyruvate. That's all. That's all. C3 versus C4. What did we learn right now? The cell type in which Kelvin cycle occurs, C3 plants I told you, Kelvin cycle occurs in your mesophyll cell normal, but here it happens in bundle sheet, the below one. C3 cycle happens in the below one here. All of you will forget, that's how to show you. C3 cycle happens in the bundle sheet here. Can you see? Right here, star mark. Happens in the bundle sheet. C3 cycle in C4 plants. Which type of cell in which initial fixation happens? Both mesophyll initial fixation. How many leaves? One type of cell, two type of cell in your C4 plants. Which is the primary acceptor is RUBP. Here it's PEP, phosphoenol pyruvate. RUBP is 5 carbon, is 3 carbon. First type of product is going to be PGA, oxalic acid. PGA is 3 carbon. That's what's called a C3 cycle. 4 carbon, C4 cycle. Rubisco, yes. Both have Rubisco. No, C3 cycle will not have pepcase. Pepcase will be only present in the case of C4 cycle because they need pepcase. They don't require pepcase. Which cell in the plant have Rubisco, mesophyll? Here in the case bundle sheet in the bottom. Why in bundle sheet? Because C3 happens in bundle sheet. Happens in bundle sheet. CO2 fixation under high light condition, medium, very high. Photorespiration, it will not happen in both the cases in the case of low light intensities, but in high light intensities, yes. Very high light condition, photorespiration will happen in C3 plant. And always remember, C4 plant it can never happen. Photorespiration never happening. In low CO2 it will happen here. High CO2 no. High CO2 is very high. There is no requirement of photorespiration. Never had happens. Optimum temperature is what? 225, 30, 34. High temperature. C4 plants high temperature. Examples wheat and rice, maize, sugarcane and sorghum in the case of C4 plants. So with that students, C4 cycle is also over, done and dusted. Now students, we'll take a 20 minutes break. I'll come back, I'll go drink some water and eat something and come. I'm a little hungry. So we'll take a break till class will resume at 5:55. Yes, 30, 35, 45, 55. Yes, 5:55 will resume the class. 5:55 p.m. 5:55 p.m. will resume the class, and then we will do the last part of today's class. That is going to be factors affecting photosynthesis. Last part of today's class. Done. Done. Okay. So we will see you exactly at 5:55 p.m. Until then, go all of you eat something and come, run and drink some water and come. All of you. Quickly go. Run. So, hello students, welcome back, and we are here to the last part of this chapter. So, I believe students, till now everything is clear, right? I believe till now everything is clear. Today we started off your chemosynthetic hypothesis, then we did your C3 cycle, then we did the C4 cycle, then we also did C2 cycle. All of these following cycles are over now, done and dusted. Now I will teach something called as factors affecting photosynthesis. Very easy to understand. And hardly you will get around one question or two questions every alternating year. Or once in 3 years, you get a question from here. Nevertheless, you can't skip it, but nevertheless you can't skip it. Okay, so let's read it. As understanding an understanding of the factors that affect the photosynthesis is necessary. The rate of photosynthesis, one second. The rate of photosynthesis is very important in determining the yield of the plants, including crop plants. Okay, it is very important in determining the yield of the plants, including the crop plants. Photosynthesis is under the influence of several factors. Internal factors, external factors. Internal factors are what are they? What are they? For example, for your ability to catch this pen. Obviously, if you have proper fingers and everything, hand you can catch this pen. What if you don't have a hand? Can you catch the pen? No. Similarly, for a plant to do photosynthesis, it needs to have large leaves, many leaves, high chlorophyll content. Yes, a good age of the leaf. When I say age of the leaf, I mean like even a moderate age. Old leaves just do not have chlorophyll and they fall off. Okay. See number, size, and age and orientation of the leaf, mesophyll cells and chloroplast, internal CO2 concentration and amount of chlorophyll. All of these are called as internal factors, and internal factors are mainly based on genetics. Mainly based on genetics. Okay. Now, what about external factors? External factors would include the availability of sunlight, how much sunlight is present, how much of water is present, how much of CO2 is there, and what is the temperature. See, obviously, students, if there is very high CO2, plants will produce more glucose. Less CO2, less glucose. More temperature, plants, all the enzymes will die. Low temperature, enzymes will not function. Clear? So as plant photosynthesis will photosynthesize, all these factors will simultaneously affect the rate. All of these factors will affect the rate of photosynthesis. Sunlight, temperature, CO2 concentration, water, all of this will work on the plants, determining the rate of synthesis. Okay. Hence, those several factors interact and simultaneously affect the photosynthesis. Many factors are affecting the photosynthesis or CO2 fixation. Usually, one factor is the major cause or is that limits the rate of reaction. Simple example, understand. You are writing NEET 2024 examination, or if you wrote NEET 2024 examination, you know that there is something which is lacking. For example, your rank was not good enough because you could not solve enough of PYQ. That is the limiting factor. If you were not doing enough of mock tests, that was the main reason that you did not score very well. You were not able to, you had a lot of backlogs. That became the very reason that you could not score very well. Similarly in the case of plants, there are many factors affecting the rate of photosynthesis. Many factors are there, but there is that one factor which is present in the less amount, one factor that limits the rate of reaction. Why it is limiting the rate of reaction? Because it is present at suboptimal levels. The meaning of suboptimal level is less quantity or less amount. For example, students, for example, understand. For example, in the winter season, sunlight will be very less. Sunlight will come very late. So sunlight becomes a limiting factor. Sunlight becomes the limiting factor. In the case of summer we have sunlight. We have sunlight. But in summertime, do we have enough of water? No. Water is present less. So water becomes a limiting factor. I believe the same concept, this same concept which you have learned in your chemistry also called as limiting reagent also. Same concept applies here. Okay. Whichever factor is present in least amount becomes a limiting factor. It is also called as law of limiting factor, also called as Blackman's law. Blackman's law. Okay, Blackman's law. Now, this states the following. If a chemical process is affected by more than one factor, sunlight, water, CO2 concentration, temperature, then its rate will be determined by the factor which is nearest to its minimal value. That is, whichever is present in the least quantity. Okay, which directly affects the process and its quantity is changed. Okay, so I believe this is very interesting to all of you to understood. Okay, how to buy the books? Watch Vedantu NEET English channel. You will get to know. Okay. For example, despite presence of green leaf and optimum light and CO2 condition, the plant may not photosynthesize if temperature is very low. Obviously, if temperature is very low, plant will not able to photosynthesis. Why? Because if Rubisco enzyme is not working properly, tell me, if Rubisco enzyme is not working properly, can RUBP take up CO2 and convert it to, can it convert it to PGA? Will this happen? Tell me. If Rubisco enzyme is not functioning properly, this will never happen. And tell me, Rubisco enzyme will only work properly when there is enough of proper optimum temperature. Because you know from biomolecules chapter, every single enzyme has a very specific temperature that is called as optimum temperature in which the enzymes work. On which the enzymes work. Clear all of you? Clear? Now, so certain factors are here. We'll understand one by one each factor. For example, first we'll understand light. We need to distinguish between light quality, that is basically some of the plants like blue light and red light, favorite light intensity and duration of exposure. Duration is nothing but your day neutral plants and everything. While discussing light as a factor that affects the photosynthesis, there is a linear relation between light and CO2 fixation rates. Now, what is the relationship? Understand. If you look at this graph, this is O. From O to A, can I tell from O to A, light intensity, light intensity is directly proportional to the rate of photosynthesis? See here, students. In this from O to A, as the light intensity is increasing, rate of photosynthesis is also increasing exponentially. Same direct proportion. Yes. But if you notice from B to C, if from B to C, no increase, no increase in photosynthesis. What does it tell you, friends? From this example, you will understand right here. At certain point, how much ever light you increase, at a point, how much ever light you increase, there cannot be any increase in the rate of photosynthesis. Understood? So you can scold someone, but at one point, how much ever you scold that person will not change. Right? Same thing is happening here. Okay. So at one point becomes saturated. After that point, no, rate will not increase. How much ever light, even if you light increase the light. Okay. That is because, students, that is because out of the total sunlight, out of the total sunlight, plants require only 10% of it. That's all is enough. Plant is happy with 10%. It doesn't require more than that. It doesn't require more than that. Just 10% is enough for the plants. Okay, see it's given here. So that is the reason at high light intensities, gradually rate does not show further increase. That is, intention other factors becomes limiting. Only 10% is required. Hence, except for the plants in the shade dense forest, light is the really limiting factor. Why? Because light cannot be limiting factor because it is not present in less. Sunlight is present in plenty always. You will not find part where sunlight is very low. That might happen in winter, but generally speaking, sunlight will be there. Okay, sunlight will be there always. Incident light beyond a certain point causes even breakout between of chlorophyll and decrease of photosynthesis. So increase in photosynthesis at very high level is also dangerous to the plant. Plants do not require so much of sunlight also. Okay. Carbon dioxide. So understand, friend, this is a very famous question. CO2 is actually the major limiting factor. CO2 is actually the major limiting factor. Why? Because the concentration of CO2 is very less. That is, in the atmosphere, it is only between 0.3 to 0.4%. That's all is CO2. Now tell me, if CO2 is present very less, obviously then CO2 becomes a limiting factor, major limiting factor, which determines the rate of photosynthesis. Okay. Increase in concentration up to 0.5 can increase the rate of photosynthesis. Okay. Beyond this level can also become damaging over a long period. So only maximum requirement is how much? maximum requirement is 0.05 for the plants. Above that, plants will get destroyed. Now, I'll tell you one more concept now. C3 plant, C4 plant respond differently to CO2 concentration, obviously. Okay. In low light concentration, not very much difference. But in high CO2 for high CO2 concentration. But in high light, where light is not a limiting factor, both C3 and C4 plants show increased rate of photosynthesis. Obviously, all of you understood that. But the interesting to note here is, compared to C4, C3 has higher saturation. What is the meaning of that? Compared to C4, C3 plants can take more CO2. See here, it has a saturation beyond C4, that is C3 plants. But your C4 plants just at 360 microliter per liter. So it tells you that if I keep a small room, if I make a small room like this, if I make a small room like this, students, if I make a small room like this, in this small room, I have very high CO2 concentration. In this small room, if I have high CO2 concentration, tell me which plant will grow well, C3 or C4? Obviously C3 plants, because C3 plants can take more CO2. Understood, all of you? And I will call this small room as greenhouse. Because that's what happens in greenhouse. In the greenhouse, there is very high CO2. So that is why, student, that is why in the greenhouses and all, you can see a lot of C3 plants growing. Example of C3 plants, very simple. Tomato and bell pepper, given right here. Can you see? Tomato and bell pepper. Tomato and bell pepper C3 plants, which can grow very high, which can grow really well in high CO2 concentration. Now, next one is temperature. All of you know, right? Enzymes are basically type of proteins, right? And proteins get denatured in high temperature. They get isolated or inactive in low temperature. The same thing will happen with all the enzymes also. All the enzymes at high temperature, right? High temperature, they will decrease the rate of photosynthesis. Yes. And even low temperature also will decrease. Okay. Temperature optimum for photosynthesis of different plants also depend on their habitat and their adapted to. Tropical plants such as your C4 plants C4 plants have a higher temperature optimum. They can tolerate more temperature. C4 plants can tolerate more temperature. Okay. Compared to your C3 plants. Okay. Next one is water. Water has rather indirect role. Complete indirect role. For example, I'll tell you. Now here I have a plant and I have a leaf here. I have a leaf here. Now tell me, if water will decrease, the leaf will show curling. Leaf will curl inside. If leaf is curling on the inside, can I tell surface area is decreasing? Tell me, if surface area is decreasing, obviously photosynthesis is decreasing. Why? Because less amount of leaf is exposed to sunlight now. If surface area is decreasing, that's all. The second one I'll tell you. Second one I'll tell you. Second one I'll tell you. This is a leaf. Okay, this is the leaf right here. If water is reducing, can I tell all the stomata will close down? If stomata is closing, can I tell less gas exchange is happening? If less CO2 or less CO2 is entering, if less CO2 is entering, can I tell less photosynthesis also is happening? So role of water is little indirect here. Okay. Rather than indirect role. Water causes stress stomata to close, hence reducing the CO2 availability. The first one is right here. Besides, water stress can also make leaf wilt, curling of leaf. That will do what? Reduce the surface area and metabolic activity as well. That also will completely decrease altogether. Let's see the summary now and let's see if we covered every single point. Now, green plants make their own food by photosynthesis. All of you know this. All of you know this. During this process, carbon dioxide, CO2 from atmosphere is taken with the help of leaf and water is added in the presence of sunlight and a pigment called as chlorophyll. They end up making the glucose and oxygen is the byproduct. This is the reaction. Yes. Principally glucose and starch. Photosynthesis takes place only in the green leaf. Why? Because of chlorophyll. Mainly the leaves. Within the leaf, The mesophyll cell have large amount of chloroplasts that are responsible for CO2 fixation. When chloroplast, the membranes are site for light reaction. Basically, thylakoid membranes are light reaction happens on thylakoid membrane. Well, chemosynthetic hypothesis pathway occurs in stroma. Okay, that is formation of ATP happens in stroma. So usage of ATP happens in stroma. Photosynthesis has two stages: light and dark reaction. Carbon fixation is dark reaction. In light reaction, light energy is absorbed and pigments present in the antenna and funnel to the special chlorophyll A, that is the reaction center, and we get the products. The two things are called as two photosystems we have P700 and P680, 1 and 2. Okay, where we get NADH formation and ATP formation in your light reaction on the thylakoid membrane. And how does it happen? With the help of chemosynthetic hypothesis. Chemosynthetic hypothesis makes up the ATP formation how by making the proton gradient, and when H plus is going to the stroma again, ATP is formed. Splitting of water molecule is associated with photosystem 2, it is photolysis of water, and oxygen is released as a byproduct. In carbon fixation, CO2 is added to an enzyme called as Rubisco to an RUBP. So RUBP, Rubisco enzyme, CO2 we get PGA, 3 carbon PGA. This then converted to Calvin cycle, the RUBP regenerates during the process of this process. ATP and NADPH are used. How many ATP? 3 ATP. How many NADPH? 2 NADPH. And sugar is finally made. Okay. Rubisco is catalyzed by the wasteful oxygenase in reaction in C3 plants called as photorespiration also happens when Rubisco forms a bond with oxygen. We can call it C2 cycle also. C2 cycle. Okay. Now some plants have C4 cycle in which we have a 4 carbon compound called as oxaloacetic acid, OAA. In bundle sheet cells, the Calvin cycle is carried out out the synthesis of photosynthesis, carbohydrate. And last is the factors affecting photosynthesis. All of you know that. Done. So with that, the chapter is over. Okay. So thank you so much, students, for today's class, and let me know if you have any doubt. Okay, all of you, let me know if you have any doubt. It could be all notes available. Okay, all notes available. Tanuja, Dikshi, all of you, let me know in the comment section how was today's class. Very simple, very easy to understand. Photosynthesis in higher plants. So I believe the next chapter should be respiration or should it biomolecules. Let me also know in the chat also later on. Okay. So thank you so much, students, for today's class, and I will see